I read the Where Christina Bowens, and I was like, "Oh, are we like the Wonder Twins activated?"
Well, because it'd be like, "We're Christina Ballins," and he would say Jordan or Jerry. Like,
Yep. Welcome to Absolute Genius, a podcast series from Thermo Fisher Scientific. I'm Jordan Ruggieri, and alongside Christina Bowens, I'm excited to introduce today's genius, Arianna Arbona. Arianna is a PhD student in biochemistry and neuroscience at Universidad Pablo de Olavide in Sevilla, Spain. Her research centers around early detection of Alzheimer's disease, and we loved learning about how she uses digital PCR and other great tools to advance her work. Thanks for joining us for this fascinating conversation. We began by asking Arianna to describe the different markers that she looks for in blood and saliva and how she goes about locating them.
So right now we're trying to find microvesicles in those fluids. So that's really difficult because we know there are microvesicles in all those fluids, but it's really difficult to isolate those. So we don't have any protocols or any idea how to do it. There are some studies doing that, but it's really difficult once you have them to make sure that you're isolating the right microvesicles. So the idea would be like in the brain, the cells are producing these microvesicles, and they might be going around in like around the body in those microvesicles. So the cells produce those, and then they secrete the microvesicles to the blood or to the saliva. And we're trying to find those microvesicles because they could have important or some information regarding what is happening in the brain. So we're trying to find some maybe proteins or nucleic acids that could give us any idea of what is happening.
What about those microvesicles are difficult to detect? Is it simply that they are there in very small amounts, or is it actually something in the structure of them that make it difficult to isolate or to discover?
The main problem would be the concentration. Yeah, it's really low. We don't have a specific like number, but it would be like around 1% of the things we have in saliva, let's say, because la saliva it's 99% water. In that 1%, you need to find maybe a 1% that would be microvesicles. And then in blood, it's something kind of the same, but the thing is that we could have microvesicles coming from all the tissues and all the cells. So the difficult thing would be to find some markers to tell you that those microvesicles are actually coming from the brain and not from another tissue or organs.
Can imagine too, you have the blood brain barrier for a reason, right? Is there some of that as a factor as well? As we know that there's a barrier from what can get in from the blood into the brain, but does it work the opposite way where it's actually even more limited of what's getting from the brain into the blood?
Yeah, yeah, that's also a thing. The good thing about the microvesicles is they're really small. So as they're that small, they can cross the barrier, and then we can find them in the saliva or in the blood. It's not completely sure that those vesicles coming from the brain are in the saliva. We have that hypothesis because we know that the saliva it's right next to all the blood system is like passing and in contact with the saliva. So as we know that, we think that some of those microvesicles could come to the saliva, and we thought about the saliva because it's a much easier thing to have from a person. So some of them don't like to have blood taken, or would be maybe a fastest way. So that's why we thought about saliva first of all, and then we try also with blood because we know there's always more things in blood.
is it very important to get whole microvesicles intact to do your analysis? Or are you just looking for, those signatures to be present, even if they've kind of degraded? I imagine, it reminds me a lot of what we talk about for oncology and circulating tumor DNA, and those pieces are just bits and pieces. So is it similar or different?
It's really difficult to know if you have the whole microvesicle or if you're not. This actually not a way to know that, because we know that those vesicles have some proteins in their membrane. So if they have those proteins in their membrane, we're trying to capture those proteins. And when we have a positive in those proteins, we say, okay, then we have those microvesicles. But you never know if you're actually capturing the whole microvesicle or they're just free proteins you have from in your blood or in your saliva.
So it sounds like on one end, you're looking at proteins in the membrane to make sure that you are actually getting the right microvesicle. But what exactly are you trying to measure in terms of an identifier that the subject might have Alzheimer's or have a pre-existing the start, right, the onset of something like Alzheimer's?
Once we have the saliva or the blood, we start extracting the microvesicles from there. And once we have those, what we're doing right now is trying to immunocapture those microvesicles. So we know certain proteins that are in the membrane, and we tried with magnetic beads to capture those microvesicles, and then we can see in the Western blot if we're actually capturing those microvesicles or not.
Yeah, no, it makes sense. And then once you know that you've captured them, what exactly are you looking for to see if they may have Alzheimer's, or even they're moving towards that, right? A predisposed, right, to kind of move that direction. Are you looking at a DNA signature or proteins within the microvesicle to help kind of see those different levels?
So the thing is that as we have the two groups, the groups that we already know that they have Alzheimer's and the group that would be not having Alzheimer's yet, we can compare once we have, let's say we can extract and isolate all the microvesicles from both groups. Then once you're completely sure that you have all the microvesicles, then you can compare the the information they have inside, either proteins or nucleic acids or whatever. So if you can compare both groups, you can see if there's enrichment in one of them in something, and then we can start studying that protein and what it does and whatever. And then we're also having a look at the proteins and all the markers that we already know that are using in other studies.
Maybe one more question about like this like kind of signature you're looking for. Like once you've identified, I have the right microvesicles, how many types of markers, both protein and nucleic acid, do you look for in that signature?
We're we're trying to find in literature and like published papers all the proteins that they have described and all the markers they have described. I mean, we're trying to find those in the microvesicles. And then we're just doing proteomics and see whatever is inside them.
So maybe if I'm understanding right, like right now we're kind of in the process of identifying that signature more than we actually know what it is, or more than one person has a signature that we're looking for. Maybe a good transition. So at the Absolute Genius, we love to talk about digital PCR. How do you use digital PCR as part of this process? And I know we talked a lot about like how we get to the cells that we're really interested in looking for, but could you give us a background on how you find DPCRs useful?
So we're not doing using digital PCR yet. We're trying to do immunocapture with PCR, so Immuno-PCR. But we're still we're exactly in the beginning of it. So we're still having a lot of problems, and as this is a new technique and no one is working with microvesicles, there's no protocol or no whatever to use. So every time we buy something, we try to do a PCR, we try to do whatever, we never find something because there's nothing published yet about this. Every time we buy something, we say, how can we use this with microvesicles? And no one knows. So then we need to run hundreds of experiments until we find the right conditions to do everything. So we do have the idea to use immuno-PCR, but we're still working on it.
That's fantastic. Yeah, you're truly trailblazing.
Another question, we've talked about this in the context of Alzheimer's. Are you doing something similar for biomarker discovery or research for other conditions?
we do know that like neurodegenerative diseases, they all share some kind of background, or they all have some things in common. So we always try to have a big view of what it could be, what we could have. So we do not just focus on the things of one disease. We usually have Parkinson's as the one we're having like most more things in common, but we always try to find things regarding the brain. So if it's a disease that affects the brain, we can always have a look and try to find similarities with those.
So another aspect that we always like to talk about here is the idea that, and it was a theme of our previous season. I'm sure it'll come out in this season, is the types of tools that you're using to accomplish your research. It's never a one fit solution. So we talked a little bit about Western blot. What are some of those other like tools and techniques that you're using to identify signatures?
So first, to isolate the microvesicles, we're using size exclusion chromatography. So we tried several types of extraction techniques, and we saw that the best one and the one we were having the best results was with the chromatography. So right now we're using that one, and then we saw that the best way to immunocapture those microvesicles was with dynabeads. So we're using this magnetic capture to have those. And we always use Western blot to check what we're having, if we're having positive immunocapture microvesicles. I think that's most of it what we're using right now.
I know at one point we discussed your work on systemic autoimmune diseases. Can you elaborate a little bit about what you're doing on the research you're doing on those as well?
So I already finished that one, and we were studying lupus. So the thing would be that we were trying to see in a multiplexed digital PCR. So we designed with Thermo Fisher the Chapman probes, and we had 4 different genes that we were trying to see in those samples we had from lupus. So we designed the probes in a way we could measure 4 genes at the same time in one sample. So the idea would be to have a faster and easier way to see if you already know, like you already have a sample and you want to test the levels of 4 different genes regarding, and that we already know they're affected in that disease. So then you can just with the digital PCR have the levels of the 4 of them at the same time.
So then maybe to also ask a parallel to your Alzheimer's research, are you looking, when you talk about nucleic acid signatures, are you talking about gene expression, so like the actual functional side, or are you looking for those markers? And I know we hear a lot about like specific like germline mutations that can make you predisposed.
We were more interested in the gene expression. So we were wanted to know the expression of those 4 genes that we already know that had something to do with lupus.
So maybe to kind of parallel, looking back at what you were using for lupus, can you describe why dPCR was helpful for multiplexing in that context, and then your future plans on how you might integrate it into the Alzheimer's research, and if there are any parallels?
So the good thing about the digital PCR, we were first trying to do it with qPCR, but the amount of DNA in those samples was so small we cannot see it with qPCR. So that's why we moved to digital PCR, because there you can see when you have 4 copies, you can already see the signal and you can already have a signal with the Chapman probes. So that's why we moved to digital PCR, because we had a really low amount, and with that technique, it could give us a really good view of what was happening, even if we didn't have enough.
Yeah, and you said something earlier that piqued my interest in your Alzheimer's when we were talking about immuno-DPCR. So are you thinking about integrating like protein detection into DPCR? Like, is that where it'd be useful, or would it be the same, like just to get better resolution for gene expression signatures?
Yeah, I think it would be useful to have better. Like if it's still the same, we're still working with a sample that has really low amounts of where you want to find. So I think digital PCR would be useful in that way, because you always can see with digital PCR things that you cannot see with other techniques. But we're still in the beginning, so we still have a lot of things to do.
It seems exciting to be at the beginning sometimes in research. It's a lot of exploration and unknowns, right? And just trying things to see if you can find some different either parallels or something that stands out that's worth investigating deeper. I actually want to go back a little bit and talk a little bit about some of your autoimmune disease research. Can you talk a little bit about some of the targets and expressors that you were looking at?
Well, with all the immune diseases, there's always like interferon signature. So we do know that the interferon signature, we cannot measure the interferon in blood. That's the thing. There are very few techniques to measure the interferon in plasma. So the idea was to use those genes to have an idea of what was happening with interferon. So we do know when the interferon increases, all these genes increase too. So we wanted to see with these genes like an indirect way to measure if the interferon was increasing, because they're still trying to find a way to measure that in blood, but there are not really good techniques to do that.
Instead of trying to measure an interferon protein in the blood, you're looking at different gene targets as a way of approximating if there would be more or less in the blood. So would you be looking maybe more at mRNA and like as it's on its way to maybe, be translated into um uh those proteins, right? You're looking more at maybe those mRNA expressors to see if they're elevated or decreased.
Yeah, we were trying to measure mRNAs. The thing is that they were really, really low copies, like really low number. So that was really difficult because you could maybe have 2 or 3 copies. So then it was really difficult to measure those. That's why I was doing this with the RNAs, because in that way we had not many, but a bit more copies. So then the PCR was a bit easier.
Yeah, I was gonna ask actually if there's any kind of special sample prep, like to bring me back on when we were looking at cell-free DNA, running the tubes from the hospital to make sure that it's, you separate everything before everything starts degrading. The half-life of RNA has to be much shorter. So how does that impact is there anything that you're doing up front to make sure that your time point is as accurate as possible?
So we were working with hospitals and we were having the samples the same day they were extracting the samples, we had them. So as we were working like that connected with the hospitals, it was a bit easier, because if you get the RNAs after a bit, like a long time, then you won't get anything. So that was the good thing, because we have connections with the hospitals. We can keep a follow up of those, and then we have the samples the same day.
That totally brings me back. So you're also running to and from, running with your ice buckets.
I've worked with RNA a little bit in my past as well, and I have to say my experience was if you look at it incorrectly, it starts degrading. So I can only imagine some of the complexity of making sure you get something that is as close to what's actually happening in the subject as possible. Can you talk a little bit again, just because I think it's interesting, and I think it would be useful for our listeners as well. What are interferons, and how what role do they play in a body?
So they're proteins there you can find in blood, and they increase or decrease depending on what is happening in the body. So when, for example, you have an infection, you have a virus or whatever, you can see the levels of those proteins that increase in your blood. And then with some other, let's say, immunodiseases and so on, you can also see the how the levels of interferon change. in the blood.
They're kind of a signaling protein, right? So they are produced when you have some sort of virus or infection to alert the body and alert neighboring cells that there is some sort of infection happening. Is that a correct assumption?
Yeah, yeah, yeah. So the cells produce that every time let's say they have an infection or they have some kind of disease that is kind of, yeah, for example, with the autoimmune diseases, you can see how the cells are producing those proteins because they do have something that is not working in the right way, let's say.
Are you looking to precisely quantify rare targets in your sample? Are you always struggling with standard curves or measuring limits of quantification? If so, Applied Biosystems QuantStudio Absolute Q dPCR system is for you.
With the Absolute Q, reliable and precise digital PCR can be as simple as preparing your samples, loading onto the plate, and running the instrument. The system consistently delivers more than 20,000 microchambers and provides results in as little as 90 minutes.
That's right, Christina. And the Absolute Q utilizes more than 95% of the sample that you input, so you won't have to worry about missing rare targets due to dead volume. You can learn more at www.thermofisher.com/AbsoluteQ or visit the Absolute Genius webpage. The Applied Biosystems QuantStudio Absolute Q dPCR system is for research use only, not for use in diagnostic procedures.
Let's go back to our guest.
Arianna, well, it's time for our career corner portion of the podcast where we talk a little bit about how you got to where you are today. Can you give a little bit of background on your educational journey and your career journey?
Yeah. So when I was in high school, I was like really interested in all the biological part. And every time they explain something to me, it sounds like, I don't know, fascinating because it seems like everything is connected. So I was thinking like, how is it possible that everything is connected in such a perfect way? So every time you touch something or you see something happening in one cell, you can see how in another cell, in a completely like separated part of the body, is also showing that something is happening. So I was fascinated by that. So I decided to study biochemistry. I did that in Madrid. And for my last year, I decided to do an Erasmus and go to Helsinki. So there I like did a lot of labs and so on, and I really liked the genetic part. So I was fascinated by the genetic part, and again how you can have just in 4 letters all that information and all the things happening just from 4 letters, if we say it in a like simple way. So then I did my master's in genetics, and I finished my master's last year, and I was kind of not lost, but I didn't know if to continue in that part because I knew that there were some other fields that I'd like to. So in the end, I end up here in now I'm in Sevilla, and I'm doing this my PhD in neuroscience. So I also like this part, and I also see how you can apply the genetics and so on in everything you work with. So it's not that I studied genetics and now I don't apply it anymore and I'm studying neuroscience. You can always mix them and trying to find similar things in both of them. So that's what I'm doing right now.
That's amazing. You talked about leaving Spain and studying in Helsinki. What was it like studying in a different country? Were there was there some really good value there that you brought back as part of your PhD? And what advice would you give for anyone that might want to study science in a different country?
So I really loved that experience. I knew and I heard that the one of the best educations we have in the world is in Finland. So when I had the opportunity to go there, I didn't hesitate. I was like, "Yeah, I'm going. I don't care." So I have to say I'm from Canary Islands, where it's usually like 20 degrees all the year. And then I moved to a country when it was minus 20. So everyone was like, "What are you doing here?" One like, seriously, what are you doing here? But I really loved that, and I learned a lot. And it was a bit different of what we're used to in Spain. So the education is, I would say it's better there. Sorry to say that, but I would say it's better because they really know how to plan everything, and they really know how to make you learn. I would say in the best way you can learn. So I really loved that, and I really loved how easy it was to, in the scientific part, how easy it was to go to a lab and start practicing and so on. Maybe sometimes here it's more like you need to have all the papers and all the things, like all the permissions to go to a lab and so on. And there it was just, I talked with a teacher and said, "Listen, I would really like to learn more about it." And he said, "Yeah, come tomorrow to my lab and we can see what you can do." So I really enjoy it there. And there were also like so many opportunities, and all the labs were super connected. And I think it was pretty easy to work there. But as the scientific part, always super connected, it's true that it's international. So you can always understand and do the things they're doing there, because that's the good part about science, that it's international, and the things you're doing here, people from China would understand it too.
Did you have any mentors along the way? It's a theme we've heard from a lot of our guests that mentors have played a really critical role in getting to where they are in their career. Did you similarly? Did you have any mentors that played a critical role?
Yeah, see, I think when I did my bachelor thesis, my tutor he was amazing, and he was always so happy. And even you made 100 mistakes, he was always so positive about it. That he also made me want to stay in the lab, because sometimes it really depends who you're working with, how it how you feel about it and so on. So I think when you have someone that is really positive next to you, things go much better.
That's great to hear, and I think a good piece of advice too. You mentioned for you as well, it was difficult to make a decision after your master's on which direction to go in next. How did you approach that problem? How did you end up coming to a decision?
So the thing is that after finish my masters, there's kind of the question if I'm gonna do my PhD or if I'm not. So you always have the pros and the cons, and I knew I really liked the lab, and I knew I wanted to maybe do a PhD because I still wanted to learn more. So that I said, whatever I hear and I feel like I'm doing it, I'm just gonna go with the flow and do it. So I heard about this project, and they talked to me and explained everything, and I was amazed about it. So they were talking to me, and I was like, I want to hear more, I want to learn more. So that was it. I said, okay, this is the one.
What advice would you give for anybody that is looking to maybe follow a similar pathway as you and get into the sciences?
I would say just to enjoy it, to stop maybe thinking about what if this doesn't work, what if I start something and then I don't like it. I think you don't need to think about it. You just need to do what you feel, and then there's always time to change things, to move to another thing. So I think it doesn't matter, even if you start something and then you see that it's not the way you want to do it, you can always change it, and of course you'll learn something from it.
That's awesome advice. I echo that as well. It's rarely a case where a decision you make is 100% permanent, right? You can always change your mind and go back and try something else. It's the backbone of even running experiments in science, right? Is you pick something, and if it's the wrong thing to pick, you go back and you try again a different way. So it's good advice. I have one more question. It's a two-parter here. We ask this of everybody. What was your proudest moment so far in the lab or in your career? And on the other side of the coin, what was maybe your most embarrassing lab moment?
I would say that would the best one that would be with my bachelor thesis because it was the first time I was kind of actually working in a lab. So when I saw that I was getting results there, I was so happy about it. And I have to say, I don't know if it was lucky or it was just that I worked really hard, but I got the results they were trying to get after so many years. And I started there and I was working with cells and we were trying to differentiate those cells. And we were doing that protocol and it took like 8 weeks and we were doing 8 weeks nothing, 8 weeks nothing. And at some point it worked and I was so happy about it. And I really loved those cells. I was like in love with them. I was like, oh, they're so cute. They're working so cute.
That's awesome. What about your most embarrassing moment?
Yeah, I think it's so easy to make a mistake in a lab. Like, I think I make mistakes every day, every day. So I remember once mm, I made a fire. So I was trying, I saw that something was burning, and then I was trying to stop it, and then I put it in the wrong place, and then everything started on fire.
Oh
And
no.
I was alone in the lab, and it was like the first week I was in that lab, and I was so embarrassed. In the end, I fixed it, and someone came and said, "It smells a bit weird, doesn't it?" And I was like, "That's it. I don't smell anything. I don't smell anything," because I was so embarrassed.
Oh, that's really funny. Arianna, thank you so much for being a part of our Absolute Genius episode today. We loved having you. It was a very, very interesting conversation. And just thank you so much for your time.
Thank you. Thank you for having me.
That was Arianna Arbona, PhD student at Universidad Pablo de Olavida in Sevilla, Spain. This episode of Absolute Genius was produced by Sarah Briganti, Matt Ferris, and Matthew Stuck, with more great conversations around the corner in future episodes. Stay curious, and we'll see you next time.